Today we are going to be talking about the T-cell receptor. Remember that you have the MHC antigen presentation homework due today by 5. A bunch of you have already emailed it to me, but if you haven't, it's due by 5. I think I've replied to everyone who sent me an email already. And in talking about MHC, remember that we were basically thinking about the antigen that was involved with T cells. And so when we did the same kind of discussion with B cells, we first talked about a little bit about the antibody and sort of the structure of the antibody. Then we had a diversion into antigens. And then we came back to thinking about the antibody and the B cell. And in particular, we talked about VDJ recombination in B cells in order to think about how that B cell receptor or antibody was made. And so we're kind of doing that same parallel process here in that now that we've talked about the antigen that is recognized by T cells, we're going to start thinking in more detail about T cells and T cell responses. So here is sort of your textbook's figure talking about T cells and T cell responses. You've got one type of T cell shown in purple. That is a CD8 positive T cell. It is recognizing a peptide that is on MHC class 1. I can tell that's MHC class 1 because there's only the one transmembrane domain and there's the little beta 2M separately. And I can also tell because it tells me at the top, but it's okay. And this CD8 positive T cell is going to potentially kill the infected cell. We also in blue have CD4 positive T cells. Those CD4 positive T cells are recognizing MHC class 2 with a peptide. And then those CD4 positive T cells are producing cytokines to influence our other cells' type of response. Of course, that is as a helper T cell. In both cases, the T cell is doing some recognition. And in both cases, the T cell needs to use a receptor in order to do this recognition. And so the specific receptor that we see in the case of T cells is creatively called the T cell receptor, or the TCR. You can see the T cell receptor on this slide. You can see that the T cell receptor in the cartoon in the middle is made of two different protein chains that are held together by a disulfide bond. They both have a variable region and a constant region. They are both made up of that same kind of general immunoglobulin-like fold. And so when you look at the end of the variable region of both of the chains, you've got 3 little loops. So it's the same idea where you have those 3 little loops from both chains coming together to contact the antigen. We have a transmembrane domain and we have only a very short cytoplasmic tail on that T cell receptor. And you can see that in a lot of ways, a T cell receptor looks like an FAB just with a transmembrane domain. And so you can sort of see all of this about the T cell receptor here. So we also have to think a little bit just about T cell receptors as receptors. And remember, of course, we talked about the B cell receptor as a receptor and its pros and cons. And in particular, one thing to be aware of about the T cell receptor is that the T cell receptor has rather low affinity for MHC plus peptide. So when we have our cell that is infected or that has phagocytosed something and it is presenting on MHC, the T cell receptor can bind, as you saw in that previous slide, but the binding strength is pretty weak. It's not really strong enough to hold the cells together too well. And this is in contrast with the antibodies, which have a higher binding strength, and some other kinds of protein-protein interactions that are much higher. So the basic idea that you need to know here is that the TCR binding to MHC plus peptide, while it's really specific, yay, it's also kind of weak. And so when we want this to happen or to work out in an actual body where there are actual cells of an organism, and these two cells are really trying to interact with one another, if we want the two cells to kind of be held close together and interact, we need a lot of other partner proteins to help with this process. And so the T cell receptor has a lot of other partner proteins that it is going to work with. You can see kind of a general example, so sort of this general overview of some of these other proteins that are really important in T cell activation here. At the bottom, you're looking at some proteins that are on the surface of the T cell. So this blue membrane is the membrane of the T cell. In the top, you're looking at the infected cell or the antigen presenting cell, whatever you want to call it. It has the yellow membrane. And so you can see we've got proteins on either side of this divide that are important for interactions. When we talk about these proteins, we're going to call some of them co-receptors, and we're going to call some of them co-stimulators. I'm going to say, just basically define those two things today. You're not going to hear about co-stimulators again for a while. All the other proteins I mentioned today are all going to be co-receptor, not co-stimulator. The basic idea with the co-receptor versus co-stimulators is that the co-receptor proteins are all helping to transduce the signal from the T cell receptor. They're kind of like all contributing to like the main T cell receptor signal. The co-stimulators are like extra side signals. You can think of them as, the icing on the cake. The things that are maybe otherwise modifying. So in terms of the actual like original signal transduction event that is happening with the T cell receptor, the other proteins that are going to be participating and making that signal work are going to be known as co-receptor proteins, while proteins that are going to be giving some other signaling cascade that might modify the original one are going to be our co-stimulators. And so this is really important to have all of these proteins together to help stabilize this interaction and make sure that the cells can work together. There's another reason why many of these proteins are important. And again, here we're really going to be zooming in on co-receptor. And so here is that view that I've been showing you of the T cell receptor again. If you remember when we talked about the B cell receptor, we talked about the fact that it had some sort of good things about it as a receptor and some weaknesses. We said that, well, it was really great because it did VDJ recombination and made this super specific receptor part, but it kind of sucked because it didn't really have a cytoplasmic region. So there was no tyrosine to get phosphorylated. There was no kinase to do any phosphorylating. We had really good receptor and we didn't really have any sort of intracellular signaling stuff. So that was a sort of weakness of the B cell receptor. So here you can see the T cell receptor. As you look at this image of the T cell receptor, what do you notice, or what might you think about the T cell receptor on those same kind of points? Yep, Miles. So it does have a transmembrane region. The B cell receptor has a transmembrane region too, but the B cell receptor then had a cytoplasmic tail that was crazy short. I think it's only 3 amino acids long, so it couldn't really do anything. So what are you noticing here? Honestly, it's equally short. So again, there is a cytoplasmic tail, but like just barely. It just like barely gets into the cytoplasm. You don't see any great place where it's pointing to tyrosines inside of the cytoplasm that get phosphorylated. You don't see any places where we're pointing to a kinase that can do any phosphorylating. You see we've got this really nice receptor that can act really specifically, but we have the same problem that we had with the B cell receptor in terms of how the heck are we going to signal with this? And so those partner proteins that I told you about on the previous slide are really kind of playing 2 roles. One of them is they're helping to stabilize the interaction between the 2 cells in order to hold the cells together, because the T cell receptor's ability to bind MHC plus peptide is pretty low affinity. But they're also providing some cytoplasmic stuff in order to help get a signal from the T cell receptor. When I think about the co-receptor for T cell receptor, there are a specific set of CD numbers, a specific set of proteins I always think about. One of these is called CD3. And CD3 is part of the T cell co-receptor. CD3 is on all T cells. So in fact, when we are doing flow cytometry, we often use CD3 as a way that we're finding T cells. And the reason why CD3 is on all T cells is because all T cell receptors have this same problem of needing a signaling partner. And CD3 serves the job of being that signaling partner for all T cells. So every T cell has to have CD3 on it, because that is how the T cell receptor does its signaling. CD3 is not one protein. CD3 is actually made of 6 protein chains. So you can see that we've got one that's called epsilon here, one that's called delta, one that's called gamma, another copy of epsilon, and 2 copies of zeta. So we've got these 6 different CD3 proteins. We've got a delta and an epsilon, a gamma and an epsilon, and 2 zetas. You can see that some of them have longer extracellular domains. Delta, epsilon, and gamma have some nice extracellular domains. Zeta is mostly intracellular. It's pretty long as an intracellular protein. Delta, epsilon, and gamma also have some intracellular region. I like this picture up in the upper right, indicating this pretty nicely. The other thing that's kind of cool about CD3 is that CD3 actually diffuses in the membrane with the T cell receptor. So I told you before that all sorts of different proteins diffuse separately throughout the membrane, and we usually have to have an antigen that pulls them together. When the T cell receptor is diffusing through the membrane, CD3 stays with it. They don't get separated. They don't have to have their proximity induced. They're always next to each other. This is actually pretty cool, and one of the reasons why I particularly like this is that the way that this worked out was actually proven by one of my grad school classmates. So what he showed is that in the transmembrane domains of the T cell receptor and all of the CD3 chains, there are charged amino acids at particular heights that actually hold the entire thing together through those charge interactions. So there are basically positive and negative charges on different amino acid side chains, sort of at different levels in the membrane, that basically hold this whole thing together and let it diffuse around together. And the key thing that CD3 does for the T cell receptor is that CD3 is providing a nice cytoplasmic tail. And it's providing some tyrosines that can get phosphorylated. So in this image in the upper right, you can see these yellow boxes in the cytoplasmic tail of CD3. So you can see that epsilon has one yellow box, delta has one, gamma has one, this copy of epsilon has one, and zeta has 3 each. Those are all protein domains called an ITAM. And you guys have seen an ITAM before. We actually talked about Ig alpha and Ig beta with the B cell receptor. They're actually have a lot of similarities to CD3. And so an ITAM is an immunotyrosine based, the base goes here, activation motif. So ITAM is an acronym that stands for immunotyrosine-based activation motif. The key is that we've got a tyrosine amino acid, which is an amino acid that can get phosphorylated by a lot of kinases really easily, and this will lead to that T cell being activated. And so you can see that CD3's contribution, when we have all of the CD3 chains together with our T cell receptor, is that CD3 is providing 10 ITAMs to this whole region. So CD3 is providing 10 different sites where phosphorylation can happen to let signaling happen downstream of the T cell receptor. And we call CD3 part of the co-receptor. So now we've got a receptor and we've got a cytoplasmic tail that can be phosphorylated. We still don't have a kinase in what I'm showing you here. When we talked about this same issue in B cells, I told you there's a kinase, just trust me, because I didn't want to get into some of the kinase details at that point. But with the T cell, I'm going to tell you a little bit about the kinase. And with the T cell, this is related to another protein that is part of the co-receptor. So the T cell receptor doesn't just need CD3 as its friend. It needs to have one other friend as well to help in this process. You actually already know about this other friend. This other friend is either CD4 in the case of a CD4 T cell or CD8 in the case of a CD8 T cell. So this CD4 protein that can bind to MHC class II, the CD4 protein is also on the T cell. You can see it's also binding to the sort of back part of class II. And it's going to be the final part of the co-receptor that's going to help out this process. While CD8 is going to be binding to the back of MHC class I and be the final sort of co-receptor with this process. So part of this is, again, in stabilizing MHCs and peptide binding to T cell receptor. By having this other protein to help out, we get stronger affinity. And the other protein varies when we're trying to interact with MHC class I or MHC class II. But the other reason why CD4 and CD8 are important is that their cytoplasmic tail is closely linked with this other protein. So you can basically imagine that there's another protein that just is always attached to their cytoplasmic tail. It always likes to hang out with their cytoplasmic tail. And that other protein is a kinase. This image shows that kinase as the kinase LCK. And everything on this is correct. If I'm being very technical, I will tell you that the kinase is LCK when it's CD4, and it's a different kinase when it's CD8, It's a kinase called FYN. In any case, both of those kinases, LCK and FYN, are in the same family of kinases. They're super similar kinases. If you tell me that it is LCK on CD8, I won't be sad. I mean, I will be sad because I get way too into this kind of stuff, but like, it's okay. Technically, the answer to what kinase is here is some kinase of the Sark family. And so those of you who might be taking cancer bio with Dr. Dunaway will definitely hear about Sark family kinases. So we've got a Sark family kinase that's interacting with the tail of the cytoplasmic tail of CD4 or the cytoplasmic tail of CD8. And so what you can imagine is that normally the CD4 molecule or the CD8 molecule is diffusing around the cell separately from the T cell receptor CD3 complex. So this is trying to show them far apart. It's hard to see that they're far apart. It's hard to make a picture where things are far apart and have them still be in the same picture. But that's supposed to be far apart right there. You can imagine really like they're like this on opposite sides of the cell. That would just be harder to draw in a picture. If, however, MHC class 2 comes around, they both bind MHC class 2. So the things that were before on other opposite parts of the cell are going to get pulled next to each other. CD4 is going to be binding to this back part of MHC class 2. T cell receptor is going to be binding to this part with the MHC plus peptide. So we're going to pull those extracellular parts of the proteins together. If we pull the extracellular parts of the proteins together, that means the intracellular parts have to come together too. We've basically forced the intracellular cytoplasmic parts of these proteins to come together. Again, this is called the induced proximity model of signaling. Now we've taken this kinase, our Sark family kinase, LCK in this case, and we put it right next to a bunch of tyrosines that are good at getting phosphorylated. We put it right next to all of those ITAM tyrosines. Here they're shown as little yellow areas. And when we put this kinase right next to all of those ITAMs, suddenly the kinase can phosphorylate the ITAMs, and we get these little pink dots, which are the phosphates. And so if we don't have CD4 or CD8 and CD3 to be partners with the T cell receptor, we can't actually get any signals through the T cell receptor. So that's kind of why they're part of the co-receptor. And when we think about T cell development, which is sort of the direction we're going, we are going to need to think about the signals that our T cells getting with CD3, CD4, and CD8, as well as other signals during development. But before we talk about T cell development, which is really sort of where we're going on Friday, we need to talk a little bit more about some details regarding the T cell receptor. And one of the things we have to think about with regard to the T cell receptor are things like, the VDJ details of the T cell receptor. So here you can see another view of kind of structural details of the T cell receptor. So you can see we've got two different chains of our T cell receptor. One of them is going to be a heavy chain. One of them is going to be a light chain. You can see they each have a variable region and a constant region. You can see we've got our three little loops at the ends, which are even zoomed in on here, that are going to actually make contact with the MHC plus peptide. This gray is the MHC and the yellow is the peptide, and this little clear blob is trying to show you the T cell receptor interacting, and you can see loops 1, 2, and 3 from each of the two chains coming together to do that recognition. And we even kind of know about the angle of the T cell receptor binding to the MHC plus peptide. So you can see all of this information here. You can also see here that the two chains of the T cell receptor are labeled as alpha and beta. You can see alpha and beta labeled here. And if you look at my previous images, you've seen those two chains labeled alpha and beta. Often, they are alpha and beta. They're not always alpha and beta. So we're going to have to talk about when they're not alpha and beta a little bit later today. But sort of the most common T cells, when you think of a T cell, the T cell you're thinking of has alpha and beta. It's like some weird T cells don't have alpha and beta. But we do have to talk about what is up with it and being not alpha and beta. So I'm showing you alpha beta T cells here and throughout and much of today, but that's not going to be the only one we're going to talk about. There was, some confusion in immunology for a while. People actually figured out the process for generating diversity in B cells and with the antibody receptors before they figured it out in T cells. So they got VDJ recombination all worked out in B cells. And then they were like, now how do we get T cell diversity? And they were a little worried because they were like, man, learning that VDJ recombination stuff was tough. I don't want to have to learn a whole second thing. And they were psyched when they learned that the T cell receptor is formed by that same process. So in fact, the fact that you've already learned VDJ recombination for B cells means you already know how it works in T cells, because it's really the exact same process. You can see that we have this DNA rearrangement happening, and then we end up with these two different chains. At the bottom, you can see the beta chain. At the top, you can see an alpha chain. You can see beta has V, D, and J. Alpha has V and J. So what do you think is another thing we could call the beta chain? Heavy chain. It's just like a heavy chain that you would see in the B cell. What do you think we call the alpha chain? Light chain. They're not like actually that different in size, but they are often referred to as the heavy chain and the light chain. Just like we saw when we thought about B cells, we've got those three little loops, those three complementarity determining regions, or CDRs. Loops 1 and 2 are based on which V segment is picked. Loop 3 for the beta chain is based on the particular combo of V, D, and J of beta. CDR3 in the alpha chain is based on the combo of V and J. So that is encoded by the junction. CDRs 1 and 2 are encoded by the V regions. So again, really similar to what we saw in B cells. And so here you can kind of see a simplified version of the alpha and beta loci. I will have to show you the more complicated version in a few slides, but for now we can look at the simplified version. You can see that, again, for alpha, we've got a bunch of V's and we've got a bunch of J's, and we've got our constant region. For beta, it seems like there was like a duplication. So there's V's and there's like two sets of DJ constant region. I'm not going to like ask you specifically about details of that, but that is, if you are looking at this slide and are like, this is what I see, you're right, that is what you see. I'm just going to call it V beta, D beta, J beta, constant beta. I'm not going to like talk about a deletion or a duplication, but yes, there is in fact a duplication there. So, this seems all pretty straightforward. This all seems really like stuff that we've seen before. But the reason why we have to spend a little bit of time thinking about VDJ in T cells is that there are a couple of places where things get a little bit different. There are a couple places where things are a little weird. And what I want to point out is that for all of these, what I hope that you realize as I'm telling you them is I hope that you realize, at least the one on the next slide, I hope that you look at it and say, oh, that's why she was being such a pain in the butt about that other thing before. Because she was going to try to point it out to us here too. And we're going to notice the compare and contrast because we learned it so well before. So there's a key thing that you want to notice on the next slide. And this is about how the 12-23 rule works in T cells. And so with the 12-23 rule, remember, we can think about the types of RSSs, or recombination signal sequences, that are on different gene segments. The V's, D's, and J's in the case of the heavy chain, or the V's and J's in the case of the light chain, to allow recombination. And I often was talking, you had to, when I asked you to draw RSSs, we always drew them as a triangle, and you would usually draw them on the inside part of a V, the inside part of a J, and then sort of either side of a D, right? Which you can see here. But when we look at RSSs and think about the 12-23 rule in T cells, or when we look at RSSs just in general, in T cells compared to what we see in antibody genes, that we see something different. So what do you notice here that's different? What might be ringing some alarm bells as you look at this slide compared to what you saw when you looked, if I showed you the same exact slide for B cells, what would look different? Abigail.
The D region and the beta chain has two different RSSs.
Right, so basically in the B cell situation, V had one kind of RSS. D had one kind of RSS. And J had this other kind. So you can see we have our two kinds. One's on D, the other one's on V and J. Now if you look at what you see in the beta chain, or in the T cell heavy chain, you see that we've got something different. It doesn't look the same. So you can see that now D has a different RSS on either side. V and J have different RSSs on them. And so sometimes some of you would draw stuff like this on the B cell problem set, and I'd be like, nope, because we need to think about sort of the comparison here. So if we look at this unique setup in the T cell, does it make sense? Can we put V with D? Can V go with D? Yeah, it's fine. V, D, here's a brown one, here's a yellow one, all good, fine. Can we put D with J? Yeah, totally fine. But there are two things that we can also think about in this conformation that we couldn't think about in the B cell conformation. So there are two possibilities that are available here that are not available in B cells. So what two things could happen here that couldn't happen in the B cell case? Yep, Julie.
In the beta section, the V and the J can combine because they're different RSS.
Yeah, so one thing that could happen is that you could skip D. You could put V with J and just skip D entirely because the RSSs go together. In the B cell situation, you could never skip D because the RSSs wouldn't let that happen. We'll come back to that. There's a second thing that this conformation allows. This one is people don't see as often. Chrissy. So the other thing is you could get two Ds joined together. So if I drew a second D here, you actually could have the two Ds go together because this D would have yellow on this side, and so you could get two Ds pushed together. So you could, instead of getting VDJ, you could get VDDJ. The second thing that Chrissy points out, the VDDJ, is called a DD fusion. And we see DD fusions in T cells. So T cells have this extra thing that happens that might give them more diversity of having a DD fusion. We do not see skipping of Ds. We don't see the first possibility, where we put V with J and skip the D, though we could, and no one knows why we don't. We have no idea how the T cell knows not to do that. It totally could. It knows it can do the DD fusions, but it doesn't do the skip, and we don't know why. Yep, Paige.
Technically, if it skipped over the D segment, is it considered an alpha?
Well, so it's on a different chromosome, so it would be a beta. To be honest with you, I think that if it tried to make a beta chain that skipped the D, it probably wouldn't be a functional beta chain and wouldn't make it through T cell development, is my guess. But I don't think we actually observed that to know. But my guess is it would just die because it wouldn't make it through T cell development. And so in general, when we think about the T cell receptor and T cell receptor diversity, a lot of it looks really similar to what we saw with B cell receptor diversity. When I was in grad school and I was TAing a version of this class, every so often we would throw one of the exact same kind of VDJ questions that you guys saw on exam 1 on exam 2 as see, it's all relevant for T cells too. I'm probably not going to do that because there's enough other stuff I can put on exam 2. But in theory, everything you've already learned about VDJ recombination is happening in the T cell as well. So we're still getting combinations of different V's, D's, and J's. We're still getting junctional diversity. We're still getting a huge number of T cell receptors through this process. And if we think about our T cell receptor, you can see if we're comparing T cells and B cells, they both do VDJ recombination. They both have some kind of heavy chains. They both have some kind of light chains. They both require RAG1 and RAG2. They both do junctional diversity and add P and N nucleotides. T cells can sometimes do this DD fusion. T cells, as we can talk about later, TCR alpha sucks at allelic exclusion. So B cells do great allelic exclusion. TCR alpha kind of sucks at it. The B cell can also secrete the receptor as an antibody. The T cell never secretes it. The T cell just keeps the receptor on the cell, and so there's no secreted version. We don't care about what kind of constant region really there is. Like with B cells, we cared if it was IgM or IgG or stuff. We don't really care for T cells. B cells make other genetic changes later in their life. T cells don't. Once we finish T cell development, the T cell is done with genetic change. So you can see a lot of similarities to B cells, a few little pieces that are slightly different, but big picture, a lot of similarities. Asterisk. Because there's one other thing we have to think about. When I talk to you about T cells throughout this semester, when I talk about T cell responses, when we think about CD4s, when we think about CD8s, we're really thinking about very classic types of T cells. Sort of the general, normal types of T cells. They are by far the types of T cells I know the most about. But there are some other weird kinds of T cells. I'm going to mention a teensy bit about them as we talk about T cell development, because where the T cell makes a choice to become a weird T cell or a normal T cell is an important point in T cell development. So that's a place that we're going to have to think about in T cell development. The weird T cells I'm going to tell you about today, when most people talk in immunology, they talk about them here, and they maybe talk about them a little bit in one other lecture. And I will also talk about them a little bit in one other lecture, but you're not going to hear about them again, really. They're super weird T cells. People are starting to know much more about them now. I would say when I was in grad school, nobody knew about them. We knew almost nothing. People know more about them, but I'm still not going to say a lot. And what I will also tell you is that there are actually many types of weird T cells. Again, they're going to get some time later in general to think about the weird T cells. You've actually already heard me very briefly mention one of them. At the very end of one of the classes last week, I told you about something called CD1. And CD1 is a protein that presents lipids instead of presenting peptides. CD1 doesn't present to MHC class 1. It doesn't present to MHC class 2. It presents to its own special type of T cell receptor that is on its own special weird kind of T cell. Those T cells are called NK T cells. And part of the reason why they're called NK T cells is because for a long time, people couldn't decide if they were NK cells or T cells. So they got named NK T cells. They are not the same as NK cells that we will also talk about later. They're their own thing. They are a type of T cell that have some NK cell features. They're just weird. So that's already one weird kind of T cell. But when we think about T cell development, we have to talk about another weird kind of T cell. So again, we're not really thinking about the classic T cells that we normally are talking about here. We're talking about this other unique T cell type. And this other unique T cell type is called a gamma delta T cell. And a gamma delta T cell is unique because it doesn't use the TCR alpha and beta chains. Instead, it uses two different chains to make its T cell receptor called gamma and delta. And so we're going to have to see what's up with gamma and delta and their recombination. And we're going to have to think a little bit about how a cell manages alpha beta versus gamma delta when it's doing recombination and when it's doing T cell development. And so if you actually look at what we see with the TCR locus, there are really 4 places in the genome where there are mini genes for T cell receptor. One's alpha, one's beta, one's gamma, one's delta. You can see that beta is a heavy chain. You can also see that delta is a heavy chain. You can see that alpha is a light chain, and you can also see that gamma is a light chain. And our cell either puts together a beta heavy chain with an alpha light chain or a delta heavy chain with a gamma light chain. So basically the cell has a couple different choices here. It does not really work. Well, beta delta doesn't seem to work. I've never heard, or sorry, beta gamma does not work. Like, you couldn't do that heavy chain and that light chain. And as we will eventually discuss, alpha delta is completely impossible. So it's only beta and alpha or gamma and delta together. We don't see other kinds of pairings. And so we have to think a little bit about what's going on here. So I'm going to show you a table from a different textbook. What I have learned throughout the years in showing this table is that it actually shows 2 things and everyone gets caught up on the wrong one. So I want to tell you what to look for, and I want to tell you very specifically what to look for. Okay? So this tells you what chromosome each of these 4 parts of the DNA are on. And it tells you in mice and it tells you in humans. I want to know what is in common in both, what is true in both mice and in humans. I don't want to know something that's only about one. What is true in mice and humans? Yeah, Abigail.
They both have alpha and delta chains in the same chromosome.
So in both mouse and human, alpha and delta are on the same chromosome. They're on the same big long piece of DNA. In both cases, it happens to be 14. We don't care that it's 14. We just care that it's the same chromosome. So you could imagine in that situation, chromosomes are pretty long. So maybe like alpha's over here and delta's over here, and they do their recombination and they're far apart and no one cares. That could be an option. That's not the option, though. It turns out that gamma has its own chromosome. So here you can see gamma. It's got some V's, it's got some J's, it's got some constant regions hanging out on some chromosome. Here you got beta, it's got its own chromosome. Here's all the V's, D's, J's of beta. And alpha and delta are actually together. Alpha and delta are on the same chromosome in the same place. So what you can see is you've got all the V alphas here, and then after you have the V alphas, you have the V deltas. And then you've got the D deltas, and then you've got the J deltas, and then you've got the delta constant region, and then you've got some J alphas, and then you've got the alpha constant region. So what you can see here is that delta is actually in the middle of alpha. Delta is between V and J of alpha. So you can see delta in the middle of alpha. This is super important for the development of the T cell. Let's imagine that we have a T cell that is developing, and that developing T cell makes an alpha light chain. When it makes an alpha light chain, what happens? Yeah, Abigail.
The delta region gets spliced out.
Yeah, delta gets deleted. So if you make alpha, you delete delta. So remember how I would make you draw stuff and show that all the DNA in the middle got deleted, all the DNA in the middle went away? Here, if I picked one of these V alphas and one of these J alphas, I would delete the entire delta locus. And so it's not possible for a cell to make alpha and delta at the same time. And so you can see here this as well. So here you can see V alpha, J alpha, and you can see all of the delta locus. And whenever we make an alpha chain, we actually delete the entire delta locus. And this is showing it in a larger scale version from this edition of your textbook. But what I want you to notice here is that the cell during development, during VDJ recombination, basically has to make a choice. Do I want to use alpha or do I want to use delta? Because if I use alpha, I'm losing my ability to do delta. And so as we think about T cell development and how T cells are going to be doing and timing their VDJ recombination, thinking about how they choose alpha versus delta is going to be one of the events that we are going to have to think about. I will say one other quick thing about gamma delta T cells, and then I will leave them until much later and not think about them again, because honestly, I almost never think about it, gamma delta T cells. Gamma delta T cells do actually develop pretty early during development. They actually are developing in a lot of cases before birth, and they seem to be largely going to barrier organs, and they have weird actions that make them in some ways act more like innate cells than adaptive cells. So sometimes they are talked about as being one of our innate like lymphocytes. So they do weird functions that are more innate like functions, and they tend to be found especially at barrier organs to help with defense at those organs, which are the places where microbes might come up first. that of course also means they're not as frequently found in the blood, so we didn't study them a lot for a long time. And so that's all I'm going to say about gamma delta T cells. From now, everything else I say now about T cells, I really mean alpha betas, but I'm not going to always say alpha beta T cell. I'm just going to say T cell because most people think about alpha beta T cells. So we are going to think a little bit about T cells now. And specifically, we're moving into thinking about T cell development and all of the details of T cell development. So with B cell development, we talked about a specific primary lymphoid organ where B cell development happened. And so where does B cell development happen? Bone marrow. Why was bone marrow sort of important for B cell development? Yeah, Paige. There's no, okay, so we're going, in a perfect world, there's no antigen, or there's no foreign antigen. There's lots of self-antigen. There can be situations where foreign antigen goes into the bone marrow. When that happens, it's catastrophe. So we're just going to imagine the happily ever after time when it isn't there. So, okay, so we aren't going to have foreign antigen, but what else is really important about bone marrow? Yep, Emily. There's lots and lots and lots of growth factors and other factors that are giving those cells taps on the head to make sure they can do development. So I told you about IL-7 and all the other like support a growing up B cell needs in order to develop well, right? So bone marrow is full of all the stuff that sort of that early developing B cell needs in order to do its development. T cells have to do some slightly different things during development. And T cells start out from a cell in the bone marrow. They come from the same stem cell that our B cell came from in development. But when that cell is going to do its development, it doesn't stay in the bone marrow. Instead, that cell leaves the bone marrow and goes to another organ where it does its T cell development, called the thymus. And so the thymus is the other primary lymphoid organ, and it's the place where T cell development happens. I often have heard of the thymus referred to as T cell college, where T cells get some special kinds of education and special kinds of training to become T cells. I also have a book that calls it Murder University, which we can come back to perhaps more on Friday. So you can see the thymus here. The thymus is not the thyroid, which is up here in your neck. The thymus actually sits on top of the heart. It has two lobes, so it's kind of like this, and it really sits down on top of the heart. And so you can see the thymus here sitting right on top of the heart. I should actually have some mice from Dr. McKittrick next week that I am probably going to get some immune cells from to put in the freezer. If anybody is interested in seeing a mouse thymus or a mouse spleen or various other immune organs, I am happy to show them to you. That's something I used to do when I had a lab section of this class, as that was lab 2. So if you guys want to see them, I'm happy to show them to you, and we can look at the thymus. And so all the details of T cell development are happening in the thymus. You can sort of see some other info here. We've got our stem cell in the bone marrow. It's going to decide that it wants to become a lymphocyte instead of a myeloid cell. And then at some point, it's either going to make the choice to become a B cell or to become a T cell. And when it sort of makes this choice and decides it's going down the T cell route, it's going to head to the thymus, as you see here. The thymus has a particularly important structure, which you can see here. So when we actually look at a thymus in, like under a microscope with histological stains, we can see two really key areas. One is called the cortex and one is called the medulla. And you can see the cortex and the medulla of the thymus here in a microscopy image. You can also see a microscopy image of it up at the top, and a cartoon image from your textbook also up at the top. I always show this version on the bottom. So I worked in a lab where we thought about the thymus a lot as an undergrad. And I knew about the cortex and the medulla, and I'd always kind of seen this version of it. And I thought the thymus kind of looked like the Target logo. Like there was one ring around the outside and there was one ring on the inside, but it's not, because it kind of shows like here is cortex, here is medulla. And it isn't just like a Target logo, it's actually like there are patches of each sort of throughout. So you can see that what it really looks like here. You can also notice that they stain differently with this stain when we're looking at them under the microscope. The cortex stains darker than the medulla does. We are going to have to think a little bit, I want to think about why that is. We're going to see a similar pattern with another immune organ later in the semester. And in fact, if like you can remember sort of what's up with the dark one and the light one, in most of these cases, it actually helps you remember some other stuff. So we're going to think about why the cortex stains so darkly and why the medulla doesn't stain so darkly. So if you were to guess, and you can actually kind of see it in these pictures pretty well, especially if you look at the top microscopy image or if you look at the cartoon. What seems to be the difference between these two areas? Yeah, Roxy.
I mean, it kind of looks like there's a little like membrane around them, the like cortex, and there's a little like thin line that goes to the medulla, that's the light pink color.
Well, so there is a little bit of some kind of dividing line, but there's not a great dividing line. But okay, what if you, again, imagine you're like a kindergartner. What's the difference between here and here, or between here and here? I'll tell you that the developing T cells here are blue. What's the difference? Yeah, Jazz.
There's just more developing T cells.
Yeah, there's more cells in the cortex and there's fewer cells in the medulla. So the cortex is darker because the light can't get through it because it's so crowded. And the medulla is kind of empty. The cells aren't nearly as dense. So again, as a preview for Friday, in one of these locations, we're going to see cell division. In one of these locations, we're going to kill autoreactive cells. Where do you think the cell division's happening? Yeah, Elizabeth. It's in the cortex. Why do you think the cortex? Yeah, it's super packed. There's all the cell division is going to be happening there. It's super packed. And so because of all the cell division, where do you think all the death's going to happen? In the medulla, because it's not so packed. And so, again, we're going to see this in some other organs as well, where there are going to be areas that are dark and light. And it's actually going to be about where's the proliferation and where's the death happening. And so that's the same kind of thing we see in the thymus. What I also want you to notice in this image of the thymus is that we've got all of these blue cells in the thymus in this cartoon, and they are the developing T cells. But you can see that they are in between lots of other kinds of cells. So there are lots of cells that are thymus cells, not developing T cells, like the structural cells of the thymus that are really key here. You can see those developing T cells are really interacting a lot with all of those thymus structural cells. Whenever immunologists want to talk about sort of the structural cells in any organ, we talk about something called the stroma, which is apparently not on here. So if I ever mention the stroma or the stromal cells, The stroma is like the structural cells of the thymus. And the developing T cells need to interact with the stroma, the thymus cells, a lot for T cell development. In fact, as you will kind of see in a couple seconds, sort of the amount of like stroma surface area is really important for T cell development. So if you have a lot of stroma surface area, you can do a lot of T cell development. If something messes up your stroma surface area, you have less space for T cell development. And so your thymus isn't going to work as well. Your thymus is kind of going to suck because you don't have very many places for those T cells to interact. On Friday, we're going to see a lot more about the specific events of T cell development. And what you can notice is that the cortex and the medulla are going to come up as being particularly important at different times for different events. And so we're going to be thinking a lot about when's the T cell in each one of these different places and what's going on. So know that these different locations, the cortex and the medulla, are important at different times in T cell development. But I've told you a little bit about the importance of the stroma and the interaction of the developing T cell with the structural cell of the thymus. So you can kind of see that interaction with the stroma here. And that is actually super important in T cell development. Part of it is just like as Emily said, the thymic stromal cells produce lots of growth factors that give lots of pats in the head to our developing T cells. So some of this is about growth factors that are made by the thymus stroma. But the thymus stroma actually does one other thing for the developing T cells. The thymic stroma is how the T cell learns what self-MHC is. So the T cell basically learns the MHC of the thymus is what self-MHC looks like. So we can actually do fancy experiments where we make the thymus genetically different than the rest of the mouse. And when we do those experiments, we learn that whatever you do, the T cells think that whatever the genetics of the thymus is self. So for example, we can take mice that don't have a thymus. So here is a mouse that doesn't have a thymus. And that mouse is of the yellow genetics. And we can give that mouse a thymus from a mouse of green genetics. We can give that mouse a green thymus, as you can see here. You can see we put it in the kidney. That is true. So there is a disease where people are born without a thymus called DeGeorge syndrome. We can cure them by giving them a thymus transplant. We actually usually put it in their quad, and it works fine. So in this case, we're going to put this genetically green thymus in this mouse. What are these T cells going to think is self? Green. So as far as these T cells are concerned, green is going to be self. They are going to be green restricted T cells. When they go out into the rest of this mouse, they're going to be like, what is happening? Everything's foreign. There's going to be some issues. So the key I want you to know is that basically the developing T cells are learning from those stromal cells that they interact with what self is. Similarly, I could take bone marrow from a yellow mouse. I could put it in a green mouse. say this green mouse didn't have any bone marrow, I could give it yellow bone marrow. What would all of those T cells grow up thinking is self? Yeah. They would think green is self, because the thymus is green. You put in yellow bone marrow into a green thymus, that bone marrow, even though it's not green, is going to be like, green is my thing. I am green. Because it developed in a green thymus. So the T cells that develop learn from the thymus what self is. And you can also kind of see this here. Here you can see if I took bone marrow from an orange mouse and I put it into either a yellow body that contains a yellow thymus or a blue body that contains a blue thymus, I can make those orange T cells think that yellow is self. Or I can make those orange T cells think that blue is self. Because they learn what self is based on the thymic stroma that they were interacting with. And so the thymic stromal cells are really important to teach the cells what self is. And so you can see, as it says on the top, that T cells are restricted by the MHC type of the thymus that they grew up in. So I want to just finish off telling you about a couple of interesting sort of thymus-related things as well. So if you ask a lot of physicians about the thymus, they will tell you that the thymus is not a very important organ in people. And they will say, yeah, they're like, they will tell you it stops functioning at puberty. In fact, if you have heart surgery, in general, your cardiac surgeon will just cut your thymus off because it's in the way. Because it really does come down and over the heart. I disagree with all of this. But what the data actually indicates is that your thymus does change in its ability to allow T cell development over time. And so your thymus does undergo something called thymic involution over time. So you can see that your thymus does make the best number of T cells. It has the best output when you are young. It does drop off over time, but it's not zero. People will tell me it's zero, and I'm like, no, it really isn't. And so it does drop off over time, and the reason why it drops off over time is because, as you can see over here in the images, you've got a young thymus in this middle image and an old thymus over here, is that actually fat cells invade, so adipose invades into the thymus, and you lose surface area. So you lose stromal surface area because of the invasion of the adipose into the thymus. When I look at mice, and when I try to get a thymus out of a mouse, or when I think about those mice that I mentioned from Dr. McKittrick, I pay a lot of attention to how old the mice are. Because as the mice get older, you can't actually see the thymus particularly well. It's really hard to see. And honestly, if I get a mouse that's very old and I try to take the thymus out, it basically falls apart in the forceps with all the fat in it when it's old. But if I want like a nice looking thymus, I need to have a younger mouse. So the thymus does decrease its output with age. But it doesn't just stop. It is going downhill. I don't think that, I think that we should not underestimate the old thymus quite as much as we do. So you can see that involution process happening here. Yep, Chrissy.
So if like a 20-year-old had heart surgery and had their thymus removed,
Mhm.
are they now like immunocompromised, or are they just fine?
So part of it would be a question of how much of the thymus was removed. How much do they have remaining? And fortunately for them, they would still have whatever T cells developed before that. And so in theory, they should probably have a pretty full number of T cells. They just might not quite have as much breadth. So the other thing I just will tell you about briefly is that, oh, sorry, Jazz?
Wait, but between 26 and 49, that's still roughly about 50% of
Uh-huh.
Sometimes make the T cells.
Uh-huh. I'm not going to argue with you. Yes.
My second question is also though, I mean, this might just be an issue with the graph,
Mhm.
but why is it such a big drop down from that demographic of 26 to 49 and then over the age of 50?
I think, to be honest with you, I don't know how well those all have been measured.
Okay.
So actually, when I was an undergrad, I worked in a lab where there were some people who did thymus stuff because we were doing T-cell stuff. I worked there from when I was 18 to when I was 22, and they took my blood every Friday and gave me $20 for beer. Whoa. Every Friday, and they actually measured my thymus output for that whole 4 years. So there is a paper of my thymus output for 4 years. And that was done, not that long ago. So I don't know how well that drop off has actually been measured out. Of course, I did not spend any of that on beer until after I was 21. Yes, Emily.
T cells are like these cells where if they find their MHC that has a pathogen, it makes the clone army.
Yep.
Does it make the clone army in the thymus, or
Nope.
does it makes it in the cell?
It makes it in the secondaries, yeah. So it's going to be the same kind of thing there.
But if like your T cell for like the flu died and you can't make a new one,
That would be a problem, so yes. So there are people, as I mentioned, who are born without a thymus. It is called a disease called DiGeorge syndrome. And it is a pretty important immunodeficiency, though we can treat DiGeorge syndrome patients by giving them a thymus transplant. There also, weirdly enough, are mice that people found that don't have a thymus. So this is not a mutation that was made in the lab. This is like a spontaneous mutation somebody found. So in fact, the mutation is not specific to the thymus. There are some other weird things about these mice as well. So mice without a thymus are actually called nude mice. So you can see a nude mouse here. People with DiGeorge syndrome have hair. It's not like if you don't have a thymus, you also don't have hair. But in nude mice, in this like natural population, this natural mutation, they both don't have hair and they don't have a thymus. So they have no T cells. They are very often used. So if I ever mention nude mice, I mean mice with no thymus, which also means they have no T cells because they can't do T cell development. They are actually often used very frequently for transplantation types of studies. I had a paper the students read, which you may read this year, where I asked why did they use nude mice for this study? And the answer is because they don't have a thymus. And the students were like, because they could see the skin transplants really well. And yes, you could see the skin transplants really well, but the point is they don't have a thymus, so they don't reject skin transplants. You may have seen a weird picture of an ear on the back of a mouse. That was a nude mouse, and the idea was that they were actually trying to see if they could grow human ear tissue outside of the human body to give people ear transplants. But people are always like, why are those crazy scientists putting ears on mice? It's because we can't develop an ear any other way. We need a full body. And it turns out you can develop such things in a nude mouse. So we will start talking about T-cell development on Friday. I don't know why there's weird noises coming from the ceiling. And remember your homework due at 5 if you haven't already turned it in.